freue mich sehr, hier zu sein, auf der gemeinsamen Sitzung der Deutschen und Brasilianischen Neurochirurgischen Gesellschaften. Ich möchte Professor Dr. Andreas Unterberg danken für die Ehre, einen Vortrag zu dieser Gesellschaft zu geben. Mein Vortrag wird mit meiner Forschung über Verletzungen des Rückenmarkes behandelt. Und now, excuse me if I switch to my native English. So, I'm going to be taking you on a journey in the next few minutes and really talking to you about what I hope will be an exciting era over the next 10 to 20 years, where we'll see the translation of many discoveries which have occurred in the laboratory and taking these forward into man. And I'm going to try to go between the clinic and the laboratory, showing you the interface between how the discoveries have been translated into the clinic and where the opportunities for future translation are. I wanted to first begin by thanking the many students whose work I will mention, the many colleagues and collaborators and the various sources of funding. And this is just a picture of our laboratory team, and sitting next to me is Professor Tatter, who's my teacher and mentor, and now my colleague for the past 25 years. And this is where I work. And all of you are welcome to visit us in Toronto. It's a beautiful, multicultural city with many German Canadians, including my parents, if you're wondering why I speak a little bit of German. And this is Toronto. This is where I work at the Kreml Neuroscience Center. This is where this is located. This is the University of Toronto system. So let's begin with the clinical scenario that I'm sure many of you have seen. Probably none of us really like to see, but the reality is that we are faced with this type of a scenario virtually every week of our professional lives. This is a young person I looked after, 39 years of age, a diving injury, C4-5 bilateral facet dislocation with an Asia A C4 level tetraplegia, a very severe injury on the MRI scan. So this is the scenario, and I'm going to use this case to really try to frame the key points of the lecture. So the first question is really a clinical one, and it's a very simple question, but it has been a challenging question to answer, and that is there a role for decompression, and does the timing matter? And I will share with you the initial results from the STASCUS trial, which is actually now completed enrollment, but the final data are with the statisticians. And will we see the translation of any other drugs, or protective therapies, or regenerative therapies, apart from methylprednisolone, which really remains a two-edged sword? And I'm going to share with you a number of very promising clinical strategies, some of which I'm involved with, some of which I will be reporting on the work of others. One of these is riluzole, a sodium glutamine antagonist, frequently used by our neurology colleagues in amyotrophic lateral sclerosis. Cephalin is a compound developed in Canada. This is a biologic, a protein molecule that inhibits a molecule called Rome. And I'll comment briefly on some other molecules. Will the promise of cell-based therapies be realized? And here I will focus chiefly on neural stem cells, although I will briefly mention other strategies. And what about the barriers posed by the chronic injury? So I'll briefly introduce this concept, and this remains a very significant challenge without question. So let's consider the mechanisms of secondary injury, and this really frames the concepts of acute management with early surgical intervention and the idea of intervening with so-called neuroprotective approaches. So here we have another young person with a C6-7 facet dislocation. This is an individual who unfortunately died from a cervical cord injury. This highlights the primary mechanical injury as well as the secondary injury caused by ongoing compression and the vascular changes in the spinal cord. This is a microangiogram of the injured spinal cord in a rat, and what it illustrates is the disruption of the vasculature with hemorrhage. This is the primary injury. The secondary injury is triggered by ischemia, which is here in the penumbra zone, around this area of primary initial injury. And then if we consider this zone in the periphery, the axons, which are here labeled in green to label a neurofilament protein, are initially intact, or almost intact, but then over time, largely due to the influx of sodium and calcium and the activation of molecules such as calpain, there's a secondary axotomy that occurs. It's a secondary injury, and this is now a human neuropathology specimen showing the cystic application, which corresponds to this area of primary hemorrhagic necrosis. The glial scar, which is deposited in the area of secondary axonal damage, and then an anterograde and a retrograde generation of the nerve fibers here, so we're left with a significant gap. So certainly a daunting challenge, but a number of opportunities if one thinks about trying to target these mechanisms spatially and temporally over time. The secondary injury mechanisms continue to be an area of active investigation, but biomedical research over the last 20 years has given us quite a good idea of the important mechanisms and their relative sequences. And this will allow clinicians and clinical researchers to really think about which mechanisms may be best to target. The key initial event is an ischemic one. So we have the vascular events that occur, reduction in blood flow, edema, a loss of all regulation. So these are changes at the macrocellular level. At the cellular level, we have then changes that occur at the cell membrane at the cellular level. So we then have ionic shifts, a loss of sodium gradient, which is a critical early event. Calcium flows with this, neurotransmitter release, and so we have glutamatergic excitotoxicity. And then occurring at variable times during the injury, we have inflammation, and then we have a triggering of apoptosis. Some of these events occur very early, and some of these events, including apoptosis, continue on for weeks, months, and sometimes even longer in certain patients. And then from a regenerative perspective, looking at this from a different perspective, and this is from a review article we published a few years ago in entitled Molecular Basis of Neurodegeneration. We have here a cartoon of the nerve cell. And here at the cell body, there's a limited expression of regeneration-associated genes. So we can think about either neuroprotective strategies to try to target the cell body response to the injury, or actually applying neurotropins. At the level of the growth cone, Cahal, of course, described the phenomenon of abortive regeneration, where the growth cone collapses in the central nervous system, so with a failure of central regeneration. And many of the molecular signals are recognized. These are triggered by various myelin proteins, most predominantly Novum, but there are other molecules. And then downstream, they signal through growth. And so there are strategies either targeting Novum that I won't have time to talk about in detail, but I will talk about one of the clinical trials that I had the good fortune to lead, which inhibited growth using a molecule called acetrin. And then there's the glial scar, which continues to remain a significant challenge, and I'll talk about cellular transplantation approaches a little bit later on. So let's now go back to the clinic. So recall the patient that I introduced you to with a C4-5 fracture dislocation. Here's another patient that I operated on, and of course this is now really a standard of care. These are not the use of internal fixation to try to produce this. The principles of decompression, realignment, and stabilization are ones that we use all the time. But the particular technical challenges of when to do this, how to do this, does it work? What's the appropriateness of the timing? These remain significant areas of controversy. So to address this, my colleagues and I have really gone on a journey, and the journey has taken far longer than I thought that this But I think we're at the stage now where we're beginning to get some answers, and certainly it's imperfect. But I will share with you some of the data that are coming out of the STASCUS trial, a collaborative effort of orthopedic surgeons, Dr. Caro in the US, and this is a prospective controlled study looking at the role and timing of surgery and cervical cord injury. These are my colleagues at a number of institutions in North America. We focus on cervical spinal cord injury in the absence of multi-trauma. So these are patients that are relatively medically stable, and we've operationally defined early versus late with a cutoff of 24 hours. We've used a number of objective outcome measures, and today I'm going to focus on the American Spinal Injury Association weighted age. We've looked at a number of other outcome measures. So the data that I'm going to present to you were part of an interim analysis that was a requirement for the Data Safety Monitoring Board to ensure the safety of the study. And this is what I'm presenting to you. And this is a summary of data that I've presented at the American Association of Neurological Surgeons. And as I indicated, the enrollment is now actually completed and the final data for the statistician. But at this time, we had enrolled 276 cases. You'll see the breakdown between early and late, and you'll see the breakdown between complete patients, about 37%, very similar in both groups, and then the proportion of patients with B, C, and D. This is the distribution of age and gender. Very slight age difference, not clinically significant, 46 versus 41. But we did account for this in the multivariate analysis, and no difference in gender. And there are other important covariates to consider: injury severity scores, comorbidity index. These are similar. This is the distribution of the baseline age examinations, all of which were done in the first 24 hours. These differences are not statistically significant, but I will show you that there are slight differences in the B's and the C's, and this was also accounted for in the multivariate analysis. So despite our efforts to have perfectly controlled groups, there are slight differences which we have made to pull apart. So I'll show you the results, or the interim results, in two ways. One will be in a table, and then it will be maybe a bit easier to see in the graph. But firstly, if you look at the patients that had a true grade change in the age of the score, we see that early surgery was associated with about 24% of patients having a true grade change versus just under 10% in the late group. And this was statistically significant. And then we computed odds ratios, so using logistic regression. And this has the convenience because we can then control for the other covariates. And then here the odds ratio is 2.8. This is the confidence interval. It's perhaps a bit easier to appreciate the results in a picture. A picture says 1,000 words. So here's the percent change on the y-axis between early and late. Early in blue, late in red. And this is on a 1-year follow-up, no improvement, 1, 2, 3 grade changes. So look, the first thing to appreciate is that surgery doesn't have a huge effect on the outcome. And that's perhaps not a surprise. After all, we're trying to optimize the outcome of people with a very severe injury. But what this does show, more clearly than the other analysis, is where the changes are occurring. It's here in the tail end, and about 20% of the patients are showing a greater than expected degree of recovery. And this is significant using the cluster regression technique. So what the interim analysis showed is, well, we looked at complications. So one of the concerns we had was, okay, great, we're seeing improved neurological outcomes, but is this at the cost, perhaps, of increased complications, which was always the fear. And it turns out, in fact, that early surgery is just as safe as delayed surgery, and in fact, may be associated with reduced complications in the intensive care units. We see 36% in the early group, 45% in the later group, and the main difference is the reduction in ventilator-associated pneumonia, which is, of course, a very important issue in this patient population. So the initial conclusions, I think, conservatively, are firstly that this is a very safe and feasible protocol. So it's certainly a very reasonable option for surgeons to consider. And there are many difficulties, hurdles from a logistical perspective in terms of trying to do this. Not so easy. The initial results suggest that there are improved rates of recovery. We'll see how the final analysis turns out. And further work is clearly going to be required to validate these initial promising results. So look, certainly not a home run, but I would argue that we're a bit further ahead in terms of the fact that we now perhaps offer a reasonable surgical protocol for teams to consider when we're managing patients such as the one that I presented. Now, what about the whole issue of neuroprotection? We've seen two landmark studies. Many studies have been done, but two of these really were extremely well done. The NASA study and the GM1 dengue study. And I won't discuss these in any detail today, but only to indicate that we clearly do need improved neuroprotective agents. And I would argue that these studies have really shown us the promise of this type of research, and certainly basic science has identified a number of clinically promising strategies. Okay, so if I had to bet on one strategy, which one would it be? It would be this one. It would be targeting sodium. And the reason is that this is arguably the key central mechanism that is triggered by the ischemic changes occurring after spinal cord injury. And sodium is key because it really regulates the biology of the cell membrane. We have an activation of various exchangers. Influx of sodium is how intracellular calcium levels rise, and it's also key to triggering the activation of ionotropic glutamate receptors, and so it's key to triggering glutamatergic excitotoxicity after spinal cord injury. Riluzole is an approved compound. I believe it's used in Europe as well as in North America for the treatment of amyotrophic lateral sclerosis. It slows down the rate of nerve cell degeneration. It doesn't work all that well, to be truthful, but it is a very potent neuroprotective drug. So these are recovery curves in animals, neurologic scores with sodium channel blockers. Here are controls. Some of these are other sodium channel blockers or vehicle control. This is riluzole. Very large differences here in neurological outcomes. And then these are the quantification looking at tissue protection. So for example, here's a control section of quite a large amount of cyst formation with riluzole. It's about half the size, and you see protection at the injury site and adjacent to the injury site. So when our consensus panel in the North American Clinical Trials Network evaluated everything, we considered a number of points. One was looking at the mechanism. We wanted to look at the feasibility. We wanted to see was this validated in other models. And the other point that was quite compelling was that this was already an FDA-approved compound with safety and efficacy in a related neurodegenerative disease. So the trial has begun to look at the safety, feasibility, dosing, and the pharmacokinetics. We have 18 patients out of a planned 36 patients which have been enrolled. We anticipate completing enrollment in this initial phase study by December of this year, and we hope to begin the phase 3 randomized trial next year. I'm not going to present any results here today, but I will say that the drug is extremely safe. It's been highly feasible, and we've had some interesting initial results. And that's really all I'm able to tell you at this point, so stay tuned. But it's quite interesting. So let's shift gears, and let's look now at a regenerative type of approach. And of course, there are many different types of cells that have been used, and I won't have time to go into the rationale behind all of these. But the one that I want to focus on today is the use of neural stem cells. And the reason I want to focus on this is arguably been one of the most promising strategies, and recently the FDA has given clearance to Geron to initiate clinical trials with a strategy that is similar to this. So it's clinically quite relevant. In the future, it will be interesting because we've had the discovery of induced pluripotent stem cells. The discoverer of this, Yamanaka, has been shortlisted for the Nobel Prize. We'll see, but it's certainly very important work. And this is the opportunity where we can potentially transform somatic cells, such as a fibroblast from the skin, and actually make an induced pluripotent stem cell. And this is the concept. In Toronto, we're using an approach without viruses. This is the work of my colleague Andrzej Nagy, who reported this strategy in Nature last year. We can introduce these transcription factors into fibroblasts. Then using what's called the default pathway, we can then induce these stem cells to make neurospheres and neural stem cells. And so the concept is perhaps easier depicted in a movie. So here we have the normal spine, and then we have the spinal cord, of course. We have myelinated nerve fibers, and these signal electrically by transmitting action potentials triggered by sodium channels in the nodes of Rahmen. The myelin is adherent to the axon by molecules such as Caspar. And then adjacent to the nodes of Rahmen, we have potassium channels. These help shape and limit the action potential. Now, with spinal cord injury, in many patients, the most common form is one where we see central necrosis of the gray and medial white matter. And I tried to show this to you earlier. In the peripheral white matter, we see delayed apoptosis of oligodendrocytes. So the oligodendrocytes die, and we have demyelination or limited remyelination. And so we lose the normal nodes of Rahmen, and we have a failure of action potential induction. And so the concept with the neural stem cells is then to use the internal scaffolding support to introduce these neural precursor cells and then to induce remyelination. And this is the concept, and the basic science behind this is really very compelling, and it makes sense. The challenge, of course, will be to select the appropriate patients to get